There is a group of bacteria which is resistant to almost all the modern antibiotics. It is a major threat to humans' life, and medical institutions around the globe have to keep doing research to form new antibiotics because of this bacteria. Hello, I am Arun, and I am presenting a lecture on group of bacteria, Staphylococcus. This lecture is part of my series of lecture on gram-positive bacteria. In this lecture, I will take you through the microbiology, epidemiology, pathogenesis, virulence, clinical features, and treatment of diseases caused by Staphylococcus group of bacteria. So let's get started. Microbiology. Staphylococcus is a gram-positive, capsulated, aerobic, facultative, anaerobic group of bacteria. It is catalase positive. Further, it is divided into pathogenic and non-pathogenic group based on coagulase enzyme that it produces. Coagulase is an enzyme that converts fibrinogen to fibrin. It is produced by pathogenic group of bacteria which include Staph aureus, the medically most important. Pathogenic group of bacteria also produces protein A, clumping factor, DNase. It ferments mannitol and glucose, and it forms golden colonies which are beta hemolytic. On the other hand, another group, coagulase negative Staphylococcus, is a less pathogenic group of bacteria that is non-hemolytic, that only ferments glucose. It does not produce DNase, protein A, or clumping factors. The pathogenic group of bacteria has around 30 species, out of which medically most important is Staph aureus, which causes a range of diseases varying from minor skin infections such as folliculitis to major lethal systemic infections such as endocarditis. Whereas cones becomes important because they produces biofilms on prosthetic valves, catheters and implants. The important species of cones are Staph epidermidis, Staph lugdunensis, Staph sphalicari, and Staph saprophyticus, while the most important medically important species of pathogenic group is Staph aureus. Pathogenesis, virulence, and diagnosis. Staph aureus is a unique bacteria that can produce both toxin-mediated and non-toxin mediated diseases. Staph aureus has a special genomic island that carries genes such as SAR and AGR, which are responsible for producing toxins such as TSST1, enterotoxin, and exfoliative toxin. TSST1, that is toxic shock syndrome toxin 1, is responsible for a deadly clinical feature which is called toxic shock syndrome. In 1950s and 60s, in some developed countries, such clinical features in menstruating women, otherwise healthy, were reported. These were fever, severe hypotension, leading to shock and death within some days. On later studies, it was found that these were using menstruating tampons, that are vaginal tampons. Staph aureus can colonize healthy vagina and remain within a parameter. It does not proliferate too much, but in presence of that tampon, these bacteria were proliferating exponentially and producing this toxin, which was later absorbed into the blood through vagina, and that lead to this clinical feature. you went to a restaurant, you ordered something Chinese, the food was served hot and it was well cooked. After some hours, you started vomiting and diarrhea and felt GI disturbance. It was due to this enterotoxin, which is a heat stable toxin, which means on cooking the food, basically on recooking the food which was kept for long hours, the bacteria may die, but this enterotoxin is heat stable and it will remain as it is within that food, and when taken in, it will be absorbed, it will stimulate the vagus nerve and start vomiting and GI disturbances. Exfoliative toxin is a serine protease that damages desmosome adherens right below our skin and causes peeling of skin. It is mainly seen in infants with clinical features such as fever, poor feeding, lethargy, convulsions, and skin abscesses, which on touching minutely will cause peeling of their skin. It can be deadly. Non-toxin mediated diseases are such as skin infections like folliculitis, respiratory tract infections, endocarditis, musculoskeletal infections, invasive CA-MRSA related infections. Once inside the skin, the capsule peptidoglycan and formulated peptidase with cytotoxins that are produced by macrophages, not cytotoxin, the cytokines produced by macrophages and endothelial cells will stimulate the proliferation of PMNS, that is polymorphonuclear leukocytes, and fibroblast, which will cause pus formation and contain the bacteria. If bacteria survives there, this capability, because of this pus formation, these bacteria are also called as pyogenic bacteria. If bacteria survives from here, it will move further to the deeper tissues. It will invade the body and start causing systemic infections that can be deadly. Yes, antibodies like IgG are produced against this bacteria, but they are not very effective. The virulence mechanism of this bacteria, which helps this bacteria in invasion and immune evasion. This bacteria, by using hyaluronidase, proteases, lipases, lipase makes its survival easy in hair follicles. Clumping factors, MSC RAM molecules for adhesion, biofilms that are produced by ICA gene. By using all these enzymes, this bacteria is capable of invasion. This bacteria is capable of immune evasion. It has a special capsule, that is polysaccharide deuter ion capsule, especially in strain 5 and 8, which prevents it from phagocytosis. It produces protein A that binds to Fc portion of IgG and neutralizes the IgG antibodies. It produces CHIPS molecules that inhibit chemotaxis of leukocytes. The special MRSA, which is nowadays causing a lot of problems. It is most resistant bacteria, bacterial species mainly. It carries a gene that is called MAC gene that provides it capability of methicillin resistance. This gene is preserved as it is within this bacteria since 1950s. This bacteria has acquired this gene from another bacteria through horizontal transmission, and that bacteria has acquired this gene from bacteriophage 8081. The MAC 1 and 3 containing strains are responsible for community acquired infections, while MAC 3 and 4 containing strains are responsible for causing hospital acquired infections, also called nosocomial infections. The good thing is that this bacteria is easily diagnosable. Diagnosis, culture can be used, and PCR techniques by taking blood sample.